Okay, so now we at least get to start on something that's living, which is going to be the cells. The cells are your smallest living unit. And the size of them, this is to give you kind of an idea of the proportion of the size. So in the middle there, you've got where most of the single cells are going to be. The scale on there, it has that human's head at 10 meters. Obviously, we don't have 10-meter tall humans. You might get a 2-meter tall human in there, but it just is to show the proportions in there. So most plant and animal cells are going to be bigger than most bacteria in there. Usually the largest bacteria is about the size of the smallest human or the smallest plant or animal cell. The human egg is an extremely large cell in there. It is almost big enough that it can potentially be seen without a light microscope if you've got really good eyes and it's stained in there. You're not going to see any detail in it, but you could go, oh yeah, I see there's something there. So I've never seen one with my naked eye, but that is what I've been told, that they are that big. My eyes are not that good, so yeah, I'm going to say, you know, maybe my eyes 20 years ago could have seen that. So because the cells are too small to be seen with the naked eye, we will be using the microscope to look at them. So in the next couple labs, there'll be some stuff that you'll do with the microscope in there. With the microscope labs, some of it we'll do in class, going through showing you how to do that, but there will be some of those that you'll be doing outside of class time, where you'll just be able to come into this room and I'll have this stuff in a box that will be labeled which part of the lab to do, because with some of them they're pre-made slides and I only have 3 slides of each type in there, so it's easier to let you guys come in on your own schedule than to try and have everybody waiting for the slides in there. I just ask that when you do it, please put them back in the containers, not randomly throwing them in the box. I get really grumpy when I have to clean up after adults, and so, you know, I don't take my mother out on you. I'm not going to take you out on my mother. Well, at least not too much on her. So that's my big request there, is just to be able to do that. With some of the things, if you do want to be able to take pictures of them, hopefully by next week I'll have stuff set up so that if you really wanted to have a picture of something that you looked at underneath the microscope, we'll be able to do that. It's not going to be efficient to have it all be that way since I only have one of the cameras yet, but maybe we'll get lucky and they'll have them in a few weeks in there. So the light microscope is going to be what we use in here. It passes visible light through the specimen and then through glass lenses, and then it will refract it to magnify it, and it will enlarge the specimen up to about 1,000 times magnification. That's the best you can get out of a light microscope in there. So what that 1,000x means is it appears 1,000 times its normal size when you look at it underneath the microscope in there. So a lot of them are still going to appear small at that. The resolution is the clarity of the image. It's the minimum distance that you need to have between 2 points where you can still separate them as 2 points. If it doesn't have good resolution in there, 2 points start to appear as 1 point in there. And with the light microscope, it's about 2 microns or 200 nanometers. It's limited by the shortest wavelength of the light used, so there isn't really anything we can do to change that. There is a visible spectrum of light that human eyes can see, and unless you get different eyes in humans, that is the best you're going to get out of a light microscope in there. Contrast is going to accentuate the difference in parts of the samples. So you stain it to create contrast between the cell and the background, but some stains will actually create contrast between different types of parts. Those are usually a little bit more complex stain than what we'll use in here, where you use 2 different stains in there, and maybe the organelles stain one color, the nucleus stains another color in there. So cell walls were first seen by Robert Hooke in 1665. And Anton van Leeuwenhoek was a lens maker that made the first microscope. And what he saw under the microscope was beasties. Yes. So I don't know, I think it's kind of cute to, you know, wash those little beasties off your hands. This picture here on the left shows you things you can see with light microscopes. So this is if you don't stain a specimen. This is if you do stain it. So the stains make a huge difference in being able to see things in there. And then we have the electron microscopy pictures over here. The electron microscope was introduced in the 50s, and it focuses the electron beams through. It will either do onto the surface or it can go through the specimen in there. In theory, you could have a resolution of 0.0002 nanometers, but reality is it still only is about 2 nanometers. But that's a lot better than 200 nanometers in there. So there's two different types. The scanning electron microscope is going to reveal the surface. And these are the ones that tend to give the pretty images that they use in like documentaries in there, because they can color them in there. And people like to see the outside and pretend that it looks like something animated and cool and moving. But you can also use the transmission electron microscopy that's going to give you the internal structure in there. So we don't have an electron microscope here. Most places that do guard them pretty carefully in there. It isn't that you can just randomly go in. Even if you go to the UW, they didn't let us use the electron microscope. I know they have one somewhere in there, but we don't even get to know where it is. And it does require toxic stains. Your sample has to be done, or dead in there. Dead, done, gone. And they have to be prepared in a vacuum. So it isn't like they are just quickly, easily prepared. There is some work in getting them prepared in there. And they're an extremely expensive tool. So cytology is just the study of cells. One thing that can be done if you want to study just a certain organelle is you can do cell fractionation, where you take the cell apart. Basically what they do is they stick them in the blender to grind them up. It's always scary when you go into a lab and you see kitchen devices in there. I just find that bothersome. But then you go through, you centrifuge them, and it will separate out the different types of organelles by size in there. So that's one way of studying them like that. So our cells that we look at, we're going to have the two major variations. We don't really get to look at archaea a whole lot. They live in environments that it's hard to get them. So most humans don't have means to go into a volcanic vent 800 feet down into the ocean. Yes, they've been studied in there, and they do get studied. There's people that specialize in studying those things, but what you will run into in most classes, unless you're specifically doing a microbiology degree and then focusing in archaebacteria for graduate work, you're not going to get to look at them. They don't live in our normal environment in there. So we'll be looking at eukaryotes and prokaryotes. So both of them are going to have a plasma membrane around the outside that's going to act as a selective barrier in there. So in this picture here, you can see you've got that phospholipid bilayer in there. So the fatty tails in the center to make it hydrophobic in there. And then the hydrophilic region on the outside where it likes watery substances, on the inside where it likes watery substances. Across the membrane, you're going to have these proteins that get embedded in there. And then you get to have these carbohydrate side chains on there. A lot of these are used to identify the type of cell it is. Inside, you're going to have the cytosol, which is a semi-fluid-like substance in there that will contain the cell components. Some of the more common organelles that you'll have in all cells, all of them are going to have your chromosomes in there that carry the genes. All of them are going to have ribosomes. Which other organelles it has will depend on what kind of cell it is. So in the eukaryotic cells, the DNA is for the most part in the nucleus, and it's surrounded by a double membrane. We say for the most part because there is some DNA that's in mitochondria and chloroplasts in there. Your prokaryotic cells, they keep their DNA in a nucleoid region, but there's no membrane around it. So it's kind of like having a big old grocery bag that you throw everything into, or having a purse that's got separate compartments in there. So you could put your DNA in the separate compartments if you're a eukaryote. If you're a prokaryote, you throw it all in the same bag and hope for the best when you need it out. So the cytoplasm is going to be the interior of the prokaryotic cell in there between the plasma membrane, or between plasma membranes. In a eukaryotic cell, it's between the plasma membrane and the nucleus. So it's basically all the stuff inside except for the nucleus in there. Eukaryotic cells are generally going to be larger than prokaryotes in there. And what's going to limit a cell's size is its metabolic requirements. The cell membrane is really important in what the cell does. So you don't want to keep growing the cell bigger and not have enough membrane to meet the needs of the inside. What that little diagram shows in there is your surface area to volume ratio in there. So with that tiny little cell over on your left, the little box in there, you've got a total volume unit of 1, and the surface area is a unit of 6. So your ratio in there becomes 6 to 1 in there. With the bigger cell, your surface area on the outside is 150. Your volume is 125. That surface to volume ratio goes down to 1.2. There's a lot more volume for that membrane to have to deal with in there. Each bit of membrane has to deal with a lot more volume. And then if you have multiple smaller cells, you can actually maintain that surface area to volume ratio that you do with the smaller cells. So when organisms get bigger, they don't make bigger cells, they just make more cells in there. So one of the things the eukaryotic cells are these internal membranes that are going to separate your organelles. It also helps to facilitate participation in metabolic reactions in there. If you need two things to interact with each other and you just throw them randomly in the same space and say, well, I hope that happens, you have a lot less chance of it than if you say, you know what, I need you two to interact with each other and I'm going to stick you in the corner so you don't really have a choice in there. And that's what the eukaryotic cell does. It makes it more efficient that way. So your genetic instructions in the eukaryotes, for the most part, are going to be in the nucleus. That's where most of your genes going to be. It's going to be surrounded by a nuclear envelope in there. It's a double membrane of lipid bilayer in there. And it's going to have these proteins in there that will go through and form pores. So if you've got membrane here, and you're going to have this pore that's going to have this pore complex in there that's going to have an inner and outer lip that's continuous. So you've got two membranes here, a membrane here, a membrane here. This pore is all continuous with it. And then on the other side, so you've got this protein here, and then you've got the membranes, the two membranes. And the pores are important because they're going to help to regulate what goes in and out of the cell in there. Most things are going to have to go through one of those proteins in the pore. Your nuclear lamina is a net-like protein that's going to be underneath in here. And it's just going to be protein filaments in there that are kind of helping to support this underneath, keep things in place. Lamina. And it thins out where the fibers in there become thinner to make your nuclear matrix in there that's going to help to hold things in place. So it's kind of like you put some reinforcements up here close to the membrane to keep that stronger. But you also are going to have some of these fibers in here so that what's on the inside isn't just randomly floating around. It's kind of like having a spider web in there to keep it from moving around too much in there. So your chromosomes, these are going to be the organized form of DNA that you use in cell division. There will be a characteristic number of them in each eukaryotic species. So we have 46 chromosomes in humans, If you end up with an extra chromosome in there, your chances of survival are not very good. Most of the time, if you end up with an extra chromosome as a human, you won't even be born. We have 46. Dogs and chickens, I believe, have 60 in there. So the number of chromosomes does not indicate the complexity of the species. They seem to be completely unrelated in there. With some plants, they can get away with having extra chromosomes, but plants are a little different about things like that. Your chromatin, this is the complex of proteins that you're going to have in the loose diffuse mass. So if we have a nucleus here, it's going to have cell division going on, you'll have a chromosome. If it's just doing its normal daily activities, you can have the chromatin that will be chromatin, chromosome in there. So chromatin only when you're dividing, because you want it all organized to separate it. Here is when you have all your stuff all spread out so you can actually get in and use the DNA. Yeah. There's not a whole lot of pictures. I'm trying to think of an example where I've used pictures, and I can't think of one off the top of my head, so it's pretty rare. You'll know if I'm going to use a picture. I don't use a lot of the pictures because they're usually an artist representation, and every artist draws it a little bit differently, so it's not the most accurate thing to memorize. So the nucleolus, it's called nucleoli if there's multiple of them, is going to be a little dense area in the nucleus that's going to synthesize your ribosomes and the rRNA. So the rRNA gets used to make these ribosomal subunits, and it will exit through these pores in here. So this is showing ribosomes going through one of the pores on here. This is showing the surface of a nuclear envelope here. An electron microscope picture. Here are the things that look like little flowers on the surface are the pores, and this is ribosomes coming out through them. This is showing where you've got a nucleolus in there, and then the chromatin being all loose, floating around in there. So the mRNA is also synthesized in the nucleus, and this gets transported to the cytoplasm so that the ribosomes can translate it and read it to make proteins. This is what you would see in the electron microscope. This is the prettied up picture that really isn't reality because somebody's drawn it to be nice and easy for us to understand. But when you look at this, you can see how it's really hard to get this out of this in there. This, you can kind of see this mesh that's the nuclear lamina under there a little bit. A lot of this stuff, like with the nuclear lamina and these things, the knowledge of that stuff is actually relatively recent in there with being able to do better stains and getting better images and resolution with electron microscopes in there. So if you're going, why didn't I learn that years ago? They didn't know. So the ribosomes are going to be your protein factories. These are going to be what carries out protein synthesis. You have a lot of these in cells, especially cells that do a lot of protein synthesis in there. So the amount of organelles you have in a cell depends on what its function is. If it doesn't make a lot of proteins, it doesn't have a whole lot of ribosomes. It just needs to be able to maintain itself. Some of them will be suspended in the cytosol, and we refer to those as free ribosomes. And they usually, one of the things we know they do is they catalyze the first steps of sugar breakdown in a cell. The bound ribosomes are going to be attached to the ER, or endoplasmic reticulum, in there. And a lot of times they get inserted into the membranes. They have more to do with helping in the packaging or exporting secretions in there. So this picture here shows some free ribosomes floating around and then some bound to a membrane in here. And you'll have ribosomes that will have large and small subunits to them. So this membrane system in there is important for helping to carry out various tasks in there. It's involved with protein synthesis. It's going to help to transport things. So if you need to move something from one end of the cell, a lot of times a membrane is a good way to do it, that you can stick it in that membrane and the membrane will act like a tube that just shoots it over to the other end in there. Well, it's kind of like more like sticking it in a water slide tube and saying, okay, we're going to send you over to that pool. Here you go. They help to move liquids, detox poisons in there, all kinds of stuff like that. So your vesicles are just membrane-bound sacs. Sometimes you're going to make substances that might be toxic to your own cell that you are. And if you put it in a sac, that's a way of isolating it so you don't poison or destroy yourself in the process. So the endoplasmic reticulum is that membrane network in there. It's over half of the total membranes in the eukaryotes in there, and it's tubes and sacs. So inside the tube or the sac is the cisternal space in there. And that being inside will be separated from the cytosol in there. And it's continuous with the nuclear envelope. So where you've got the nucleus here, what actually happens is you go on to make this endoplasmic reticulum. So that if you do make something in here that you don't want to just send out through the pores, you can send it out through this tube to get it places as well. It's a way of guiding it there a little bit more. So we use the smooth and rough ER for different things. Smooth is the one that lacks surface ribosomes. It's more important for lipid synthesis. So when you think about it, membranes are lipids in there. That would make sense. It's going to be involved with making steroids and phospholipids. They'll be involved with detoxifying drugs in the liver. The way you detoxify things in the liver is if you add a hydroxide group to it in there, it increases the solubility so that you can flush it out of your body and urinate it out. So if you take in toxic things, a lot of times the urine will smell different in there as a result of it because the liver's put a hydroxide on it. Now it's water soluble and it will leave in the urine down there. So if you have a lot of proliferation of the smooth ER, it can end up increasing a person's tolerance to certain drugs. So some drugs are going to be processed in the liver before they actually act in the body in there. And so when you do that, a lot of times you have to take more and more and more of the drug because your body is adapting to it in there. In muscle cells, they're going to store calcium ions in the sarcoplasmic reticulum. You need the calcium ions to be able to contract muscles. So the rough ER will have ribosomes on the surface in there. This will be more involved with protein secretions in there and making glycoproteins. The glycoprotein just has a carbohydrate and a protein in it. We'll make secretory proteins and help keep them wrapped up from the cytosol in there. Also, it will help with some of the membrane synthesis. Yeah, but at least this is alive. It's a little easier to care about something that's alive. So this is just showing how you can have all these membranes in there and you can kind of pinch it off with stuff in it to make a vesicle. So here it's starting to make a vesicle in there. So it's kind of like if you think about gum and somebody sticks their tongue in it and it wraps the gum around their tongue in there. Then if you cut off their tongue, you could seal off in a little wad of gum and, I don't know. Kind of a morbid thought, cutting off the tongue, but most of them are going to, they aren't necessarily going to have direct physical connections. They're going to be touching each other because there's no space left. But they will usually have proteins on the surface in there that are going to interact and help communicate with the neighboring cells. So you don't have many cells that operate completely independently in your body. Where they're connected, yeah, everything in the body is pretty much connected like that. We tend to teach their functions as if they operate solely, but then we throw you out into the field and have you go into healthcare and you realize, oh my God, it's all so tied in together, and no one taught me this. So getting those to connect in your brain, it really happens just as a matter of using the information. There really isn't anything that you can do to study it. You just have to use it and apply it, and you build the connections. So the Golgi apparatus, this is a flattened sac of membranes in there. Inside, you've got the cisternae in there. These will help to modify and store proteins and then send them to other destinations. So sometimes you don't want to make things be completely ready until the last minute. This is a way of getting it mostly made and just putting on the finishing touches before you send it out. So this is an electron microscope picture of a Golgi. This is a drawing that's been made nice and pretty of a Golgi apparatus. And they have an in and an outside. Basically, they have a cis and a trans face in there. So you'll have the cis face where things are going to come in. You're going to receive the vesicles. And it will give rise to the trans side where the vesicles will exit. And in the process of going through, they're different. Did Ed leave a lot of biohazard stuff? Okay, I'm trying to train him to take his own garbage out. So we need to find out from Becky where that is. And me.
It's downstairs. I showed him where it was last night.
Oh, you did?
Yeah.
Did he take it?
No, because we didn't have it. I don't know if we have a container for it.
He could take it in the biohazard bag and deposit it. I know. I'm
And your adapter
is not over there.
There's something hooked up to the microscope in the lab now.
That's not that one.
The only adapter missing were to Wayne.
Okay.
That's it.
So anyway, with the cisternal maturation model, it goes in one side, comes out the other, and it's different on the other side. A lot of times the Golgi is going to put an identification tag on it that says, take your garbage here. So lysosomes, these are membrane-bound sacs in there. A lot of times they're going to have hydrolytic enzymes in there that will help to digest other macromolecules. So lyse means to split something open. That's what these enzymes are going to do. So some ways that we use those with phagocytosis, if you have a food vacuole where you have taken in some food into the cell in there, it will be in a package. That package will fuse with a package of lysosomal enzymes, and that will dump those enzymes in there to break down that food in there. And then you'll have it broken down into the monomers that you get to use to build whatever you need. Sometimes the cell will do this on itself, autophagy, where it will use it to recycle its own materials. Can you think of an example where you might do that? It's in more detail than 118 was. But yeah, same ideas of phagocytes and lysosomes. Yeah. So any idea where you might use some autophagy? Can you think of anything that you've had in your body you want to get rid of other than fat? Yeah, it doesn't work on that. If you figure out a way to make that happen, let me know. So one of the things is you get injured and you make wounds and they have pus. No one wants to keep pus forever. So your body actually breaks it down and recycles it. It's kind of like if you were to take and eat your scabs. You'd recycle it in there. Yes. Yeah, so you can, if you want to try to remember it by combining it with like my dog goes in the yard and he's a Phagofaecalis beast in there. The cellulitis is actually more the result of an infection in there. They will create pus from that infection, and the body's doing autophagy to try and keep that pus from completely taking over their body. But in a lot of cases in there, they'll build more pus and inflammation as a result in there. But anytime you have inflammation in your body, you don't want to keep it around forever. Once it's no longer needed, you'll do autophagy to kind of break down and recycle the stuff you no longer need. Yeah, the immune system doing autophagy and cleaning that up is what makes it go back down once the cause that is creating it is removed. When you reinfect in there, that it's never actually eliminated the infection. Yeah, it's just it building back up. The infection's actually not eliminated in there. So the cause of it's not been eliminated. It just keeps coming back. But it's harder and harder to eliminate infections these days. Sometimes you just can't get it out of the tissue and you have to cut it out. But if it's in your face, you don't want to cut out tissue. You don't really need to either. So with Tay-Sachs disease, this is an example of being missing a lipid digesting enzyme in there. So this is missing some of the lysosomal enzymes, and it allows the lipids to accumulate. It's a rare disease, but it allows it to accumulate in the brain and will cause severe problems, ending up being fatal for the person. Just want to take a look and see how far this section goes. Yeah, we're not going to finish it, but there'll be a little bit you can look at home. But we'll get through the majority of it in there. It's actually easier for me if I can have both of the sections of the class end at the same place. Then I'm not going, what did we do? The other group is all high school students. So food vacuoles, these are going to be formed by doing phagocytosis of food. So when the food comes in, you take it into the cell, and you're going to wrap it up in this package. It doesn't just go randomly floating inside the cell, because your body needs to know, okay, what is it in there? And that way you can send enzymes for protein in there. If it's a protein, you can send enzymes for carbohydrate if that's what it is. Contractile vacuoles, these pump out excess water. Some of the freshwater protists need to have really active vacuoles in there. If you are an organism that lives in fresh water that doesn't have a lot of solids, but you have a lot of solids on the inside, diffusion is going to cause all this water to keep coming into you. They have to have a means to pump it out, or they'll just swell up and burst in there. That's similar to what happens with edema in a cell, but with a protist, they're just a single-celled organism. So if you were to take some single-celled organisms that live in the ocean, and you just throw them in a bowl of water, you'll usually kill them. They'll just swell and burst. So central vacuole, in plants they have this. It's a coalescence of smaller vacuoles in there that will hold reserve ions in there, helps to dispose of metabolic byproducts, contains pigments, so your chloroplasts are one of those. Some of them will contain poisonous compounds in there. So if a plant has something that's poisonous to try and discourage predators from eating it, this is where it will keep it so that it doesn't poison itself. But if you eat the plant, you eat the vacuole in the cell and get poisoned. So mitochondria and chloroplasts, these are sites of metabolism. Mitochondria does cellular respiration. Chloroplast does photosynthesis. They're enclosed by a membrane, but these aren't actually part of the endomembrane system in there. These membranes get made by the free ribosomes, and these organelles are going to have a small amount of DNA. So the way that these organelles are thought to have arisen is that you had prokaryotic cells, and what happened is you had one prokaryotic cell engulf another prokaryotic cell, and it didn't die when it got eaten. Instead, it lived inside in there. They say that's why it's got a double membrane around it, and it's got its own genetic information in there as it got eaten and it didn't die. And you had some that did aerobic respiration, and those are the ones that became the mitochondria. Some that did photosynthesis, and those are the ones that became the chloroplasts in there. So what's kind of interesting with this, with the mitochondria and that DNA in there, is when you are conceived, you have mom's egg that has her mitochondria in there, and you have dad's sperm that comes along. Dad's sperm drops off genetic information, but that's all it drops off. So what you actually inherit is your mom's mitochondrial DNA in there. So, and the mitochondrial DNA, one of the things they think it's responsible for is metabolism. So, yeah. So I look at that and I'm like, oh, there is not an athletic bone on my mom's side of the family. Large line of large women for me to take after. You inherit a little bit more from your mother's side. Yeah, there's some things in there. They think that there may be some ties with Alzheimer's in there. And I'm like, great. Mom's side of the family's got Alzheimer's for me, too. So, you know, if you find me wandering the hall and I don't know who I am, we know that it's finally taken over. Give me cookies and let me go play in the woods and be crazy. So the peroxisomes, these are oxidative organelles. They aren't part of the endomembrane system. What would you expect to find in a peroxisome? Peroxisome. So if you look at the name, hydrogen peroxide in there. So we'll go ahead and stop there. If somebody wants to make a note, we were on the mitochondria here. Well, it will come up as 9 of 66 on here.